Okay, awesome. So now we're recording. So again, thank you for joining me. By profession, please mute yourself, guys, because I do not have now a capability to mute you. So please courteously mute yourself. Otherwise, my slides are going to be jumping. I am a very proud life manager, and again, a very honored member of Scientific Advisory Board. We started LifeVantage Doctors Group with very, very few people, and now we are massively expanded to well over 1,000 participants. So, and there are so many questions about NRF2, NRF1, and NAD. By no stretch and by no means I consider myself an expert in all of this. No, no, no. I learn exactly the way you learn, and I accumulate my knowledge from reading, and I just love teaching and I love sharing the knowledge. And you know, I don't know how big of an expert I am, but whatever I've accumulated, and I also try to make sense of what we have and explain that tremendous benefits of NRF2, NRF1, and NAD per tandem in terms of cellular machinery, biomechanics, biochemistry, and what does it do for our cellular health. By no stretch per se, I'm going to be discussing treating, curing, and mitigating diseases. No. Today, what I'm trying to do is to review that progression, why actually it is so important for us to implement that Trisyndergizer. Why does it make sense to me? So let's start with my favorite thing. And of course, I need to brag and show you the picture of me and Dr. Masayuki Yamamoto. Dr. Masayuki Yamamoto, he did not discover NRF2, but might as well we can say that he has discovered, he definitely cloned NRF2. It was cloned in his laboratory. Dr. Masayuki Yamamoto is absolutely the godfather of NRF2 and front runner of every NRF2 scientific endeavor. He is the biggest guy. I respect him absolutely tremendously. This is from the picture from Japan. I attended the conference in Japan in September, NRF2 conference. So what I wanted to start is, now, obviously, this slide, actually, I'm not supposed to show you any slides from the conference, and that's why I'm not showing you any reference or the name or anything, because I was not able to find the analogy of that slide. To me, this slide is so important because it shows the cellular progression from normality and our physiologic response to cellular stress to the severe oxidative stress that eventually is going to lead to the disease. But what you see here, the involvement of cellular machinery and those transcription factors that we know, so starting with NRF2, you know, in mild oxidative stress. And then adding, for example, mitochondrial biogenesis. Here they don't, you know, the author, it doesn't show us NRF1, but PGC1 alpha and NRF1, they are a brother and a sister, a brother and a brother, such participatory element in mitochondrial biogenesis. And then we start dealing with bad actors like hypoxia-induced factor or NF-kappa B factor, or our savior P53 tumor suppressor gene, or something that implement a cellular protection of apoptosis. And if there's a failure, you will see of P53, bad things are happening, including cancer. Absolutely. Now, another thing about this Zoom is, guys, I'm not diluting any medical jargon, any medical language, because every slide that is used at this Zoom is either from a medical paper or from the conference. So this is peer-reviewed, absolutely. Now, NRF2 and KEAP, we know that NRF2 is that latch at hinge complex. That is exactly what this slide represents. NRF2 is not just a simple protein. The more I learn about NRF2, the more conferences I attended. I have attended by now 3 NRF2 conferences. The more complex it becomes in the morphology or the foundation of NRF2, how it becomes more complex in the science, but more and more it gets into understanding of the functionality and NRF2 in association with different conditions and different diseases. So latch and hinge is between NRF2 complex and the KEAP1 complex. And there are 2 very important parts that are latching and hinging. NRF2, you see, is latch and hinge on 2 KEAP1. And it is via DLG in motif or part, and the ETGE or edge motif or edge part. It comes very, very important when we talk about the functionality of NRF2. This is another part that we wanted to discuss. Guys, there are 2 major pathways in NRF2 upregulation. The one pathway, which is called physiologic pathway or basal regulation, which is turned on by the reactive oxygen species or chemoprotective agents, which are called NRF2 activators. And out of this chemoprotective agent, we can put NRF2 protandum. From the basal regulation, how does it go? As you see here, when there is that stimulus with very mild volume of free radicals or a mild volume of oxidative stress or proper NRF2 upregulation, there is a disassociation of NRF2 from that KEAP. So you see NRF2 is disassociated, becomes on its own. And what it does then it goes and induces what is called ARA or antioxidant response element. And that antioxidant response element is going to turn on those famous survivor genes. And at the conference, the number of genes that was mentioned was 500 survivor genes on NRF2 upregulation for now. And it is done, as we know, in that basal regulation. When the job is done, what happens is, then there is, please mute yourself, then there is degradation or what is called ubiquination of NRF2. That is the safety of the basal pulsatile manner when it goes up and down, up and down. And see, that is why it is called homeostasis. It's a normal regulation of our cellular metabolism. The second pathway, as you see here, it's called constitutive pathway or hyperactivation of antioxidant response element-driven genes when KEAP and NRF2 are disassociated or disembark constantly with the persistent activation. It is abnormal. This is what cancer acquires. And this is what proper NRF2 upregulators do not do. Physiologic NRF2 or chemoprotective upregulators like NRF2 protandum, they do not have the ability. They have the ability to do this, and then NRF2 is degraded, and goes back into that hinge and latch complex. Now, Dr. Ken Ito. Dr. Ken Ito is the guy who was a postdoc in Dr. Masayuki Yamamoto lab. He cloned NRF2. NRF2 is so I quote him, NRF2 is a transcription factor that activates cytoprotective genes. I'm just summarizing. On stress situation, KEAP1 efficiently degrades or ubiquinates NRF2 and leads to proteasomal degradation. In a stress condition, KEAP1 is inactivated. That's via that mild ROS accumulation, and which leads to NRF2 stabilization and upregulation of ARE. NRF2 target genes, and you see some familiar actors like glutathione transferase reductase family, NQO1. Now look, now we're already tying up NRF2 and NAD because NQO1 is going to upregulate NAD, NADP, and NADPH. Antioxidant stress enzymes and proteins like sulfur-redoxins, pyridoxy-redoxins, thyroredoxins. Again, we also know the connection of thyroredoxins to NAD. Glutathione biosynthesis enzymes, NADPH synthesis, and we will talk later about pentose phosphate pathway enzymes. These are the enzymes that are so critically important in the mitochondrial functioning, into mitochondrial production of ATP, aka energy. Heme and iron metabolism, our famous hemoxygenase, ferritin, and other transcription factors. 3 ways that NRF2 inducers improve diseases: antioxidant function, anti-inflammatory function, and metabolic reprogramming. That was so important to me. I wasn't familiar with metabolic reprogramming, and that's what I've learned in Japan. Cancer. Cancer cells hijack NRF2 for malignant growth. There is a mutation either in KEAP1 complex or NRF2 complex, or in interphase in many types of cancers. These mutations activate NRF2 in that constitutive manner. NRF2 activates cellular defense enzymes in the cancer cells to acquire that cancer cells radio or chemo resistance. And when I showed you that ETG motif or DLG motif, where the NRF2 is latched or attached to KEAP1, these are the ones, these are the mutations. NRF2 activation in tumor microenvironment. That is so important to me as the cancer doctor. How can we upregulate tumorous microenvironment and suppress the tumor growth? So NRF2 activation in microenvironment suppresses progression of what is called NRF2 addicted cancers. And I think that's where the medicine and treatment are going to be facing. There will be 2 ways. A, how to act on this NRF2 cancer cells mutations, how to downregulate them and suppress them, but how to upregulate the NRF2 in the microenvironment. And I honestly believe that you can only upregulate the tumorous microenvironment through that pulsatile NRF2 upregulators. Oh, this is just my thought. And this is the summary. So that's what I was telling you. Somatic mutations of NRF2 are localized right here. And they're localized right here where NRF2 is attached to that KEAP. So then there are mutations, so there is no attachment. So NRF2 in cancer cells is loose and always upregulated. I guess I'm boring someone. People are yawning. Now, so this is the summary. Don't. That's okay. I'm just making a joke. It's in a live presentation. So now, activation. So now I just summarize a few notions. Activation of NRF2 suppresses autoimmune arthritis. NRF2 is protective not only oxyphilic stress, which is our famous reactive oxygen species of free radicals, but it is protective against electrophilic stress. Think of your cell phones. Think of all that electrophilic physical stress in the cell. Genetic activation of NRF2, this is so important because look, that's what I've learned. Genetic activation or proper activation of NRF2 causes longer small intestinal villi. Do you know what that means? It means that if we turn on properly NRF2 in small bowel and elongate those villi, we are improving the digestion and the absorption, because where does digestion and absorption happens by and large? In the small intestine. NRF2 may affect enterocyte or the small bowel cell or colonocyte lineage by a crosstalk between signaling pathways in stem or progenitor cells. So important just to get it right there from the start. This is, again, now I'm trying to link NRF2 to the mitochondria and NRF1 and NAD. So NRF2 affects mitochondrial function in part by increasing fatty acid oxidation. And now here, guys, it's a very busy graph, but what you can see here, something familiar, NADPH. You can see something familiar as NADPH, upregulating glutathione. So that's why it is so important to take it per se in a complex, in that togetherness. Mitochondrial aging. From one of the readings, in humans, ATP producing capacity is decreased by 8% per decade. That is why we need to boost mitochondria with the quality and with the quantity. Elderly people were found to have a 1.5-fold reduction in oxidative capacity per mitochondrial volume and 1.5 reduction per muscle volume. This was a human study. Why muscle volume? Because through the skeletal muscle, they were able to measure the number of mitochondria. So sorry, now my husband is walking in. He's going to walk into the room. Mitochondrial dysfunction. Mitochondrial dysfunction, including decrease in oxidative capacity, increased oxidative damage, is thought to substantially contribute to biologic aging. And one concept considers aging as a result of accumulation and of damage to biomolecules via reactive oxygen species. So now again, I'm tying up NRF2 activation and now NRF1 further on and NAD. According to the theory, with age, mitochondria accumulate reactive oxygen species, induce damage, and become dysfunctional. NRF2, NRF1, and NAD importance. Moving to another topic. Defective mitochondria fail to function properly and have an impaired oxidative capacity, skewed towards increased ROS production. Now, here comes the concept of mitophagy and autophagy. That is where the mitochondria in the cell itself actually discovers and identifies the bad cells and tries to destroy that mitochondria. And then from that mitochondria destroyal, then there will be cellular wipeout. Through, again, that's why the cellular functionality and complexity, please mute yourself, whoever just joined, through the lysosome or through the another lysosome is our garbage bags. to degrade functional mitochondria through fusion with lysosomes and then removing them from with the bad cells. With age, mitophagy was observed to decline because the number of dysfunctional mitochondria becomes bigger and the number of functional mitochondria becomes smaller. And there is the gradient. There is a ratio per cell when the dysfunctional mitochondria wiped out. So it has to be no more than 2/3 of dysfunctional and 1/3 of functional. That's how much we actually can abuse the cell. But when that ratio is disrupted, the cell is not recognizing that mitochondria, and then neither mitophagy nor autophagy is happening, and bad cells are starting to accumulate each other. So mitochondrial quality control and balance between mitochondrial biogenesis or synthesis of new mitochondria and mitophagy is critical, as I mentioned previously. The turnover of damaged mitochondria helps to prevent excessive generation of reactive oxygen species, which protects, look, the nuclear and mitochondrial genome from DNA damage, thus preventing DNA mutation and promoting cell survival under energetic stress. So now we're leading everything again into preventing mutations, which we obviously do not want bad mutations because they cause cancer. NRF1. Now, NRF1, I have the things that the Zoom part is interfering with my slides, and I don't know how to move it. NRF1 is a transcription factor. And what I have discovered through research, NRF1 transcription factor actually targets over 11,000 target genes, even more than NRF2 nuclear transcription factor. It regulates cellular growth. It activates nuclear genes required for respiration. NRF1 upregulates heme biosynthesis. It controls mitochondrial DNA transcription and replication, which is called mitochondrial biogenesis. NRF1 has also been associated with the regulation of neurites outgrowth. And it is so important because further on, I will show you the connection of NAD to our neurons and to our sleep pattern. How NRF1 pretandem works. Increased expression of NRF1 gene and either genes required for the maintenance and function of mitochondrial respiratory apparatus, mitochondrial DNA and mitochondrial production through the pathway independent, or they go together, but it's an independent pathway from another mitochondrial biogenesis pathway, PGC1 alpha, ultimately to give us energy. This is a study that was performed, and that is to safety of NRF1 pretandem. To me, this very important. This is a very important slide, which shows, again, NRF2 pretandem has a basal pattern of upregulation, pulsatile, up, down, up, down. Same thing with NRF1 pretandem. As you see, in 6 hours, there is 60% of upregulation of NRF1 via NRF1 pretandem Trisyndergizer. And at 24 hours, the upregulation is back to the basal state. This, to me, is the safety. The question about NRF1 and cancer. So this is a quote from the paper, Estradiol and tamoxifen. So estradiol is the female, and this is about synthetic estradiol, not us-produced estradiol. Tamoxifen is the anti-estrogen drug used in breast cancer. Estradiol and tamoxifen regulate NRF1. And mitochondrial function in mouse mammary gland and mouse uterus. So this was done on cancerous murine cells. And you see, nuclear respiratory factor stimulates the transcription of nuclear encoded genes. Again, it's a different mode of upregulation. That's in, but, and then look, estradiol and tamoxifen upregulates that NRF1. It's already done. It's already in that cancer cells. So, and estrogen increased NRF1 protein in the uterus and and the mammary gland cells. However, in tamoxifen, it increased NRF1, but did not increase the protein expression. Again, which gives me some sort of a nice safety, warm and fuzzy feeling. If you need the paper, I have the full copy. Cancerous mitochondria. In order to survive periods of metabolic stress, cancer cells must be able to sense and respond to dramatic shift of nutrient availability in their environment. So the mitochondria is a key element in our health, but the mitochondria is a key element in cancer cells as well. It's a key component of adaptive activities that not only participate in their glycolysis, in their oxidation of glucose, but also in the oxidation of fatty cells, glutamine, lactate to satisfy bioenergetic and biosynthetic needs of cancer cells. So the NRF2 upregulation of cancerous mitochondria it's a different pathway. It is driven in a different way, in a different direction. Trisyndergizer, why? Genome instability characterized by an accumulation of oxidative DNA damage, combined with a decrease in DNA repair capacity, that leads to an accumulation of mutations that initiate tumorigenesis. We need to repair dysfunctional cells. If cells persist in a dysfunctional state, they could initiate maladaptive metabolic switch towards the utilization of glucose and upregulate NAD synthesis pathways, which initiate a series of incompletely understood events that may initiate tumorigenesis. Again, it is incompletely understood, and it is totally incompletely understood by me. So I'm just learning. I just know that we need functional and healthy cells. NAD. NAD is an essential small molecule in every cell. And it's a cofactor in metabolic redox reactions carrying high energy electrons to give us ATP. By reversibly oxidizing or reducing NAD, that's why there is NAD and then NAD+. Now, that links cellular metabolism with epigenetic regulation and DNA damage repair. That is why we need everything. NRF2 upregulation, NRF1 upregulation, proper NAD upregulation. These protein modifications contribute to the maintenance and regulation of chromatin structure, DNA repair. Now, circadian rhythm. Circadian rhythm means our sleep pattern. And we know that the healthy sleep is absolutely critically important to cellular health. Metabolic responses and lifespan. That is an importance of NAD. NAD metabolic decline, one hallmark of aging, can lead to different things, as you see here. And the central metabolite, nicotinamide adenine dinucleotide, is emerging as an important aging metabolite that may be a common link between age-related genome instability, metabolic decline, and associated comorbidities such as diabetes, neurodegeneration, and cancer. Please excuse me for a second. I'm just going to close the door. So sorry about that. Mammalian cells make NAD by 3 different pathways. A, de novo, from different precursors, generation from nicotinic acid through very fancy pathway, don't ask me about that, nicotinic acid, vitamin B3. Synthesis from nicotinamide or nicotinamide riboside via salvage pathway, via what we have in the cells. I think this is important for understanding. Now, NAD metabolism is a dynamic cycle that functions to shuttle, as we mentioned, electrons to maintain redox homeostasis and bioenergetics. De novo synthesis, however, accounts for a minority of total NAD pool, while the majority of NAD comes from recycling of NAD breakdown product, nicotinamide, by salvage pathway, different kind of enzymes that are very important. It is very complicated, but I needed you to know that. Now, abnormal NAD metabolism, which is called aberrant NAD metabolism, is associated with a number of diseases. And usually aberrant NAD metabolism comes with the NAD deficiency. NAD also serves through different biochemical reactions. It serves as a substrate to very, very important molecules like PARP molecule, sirtuins, and other ones, as you see, NAD, CD38, CD157, that is easier for me to pronounce. Thus, NAD regulates DNA repair, gene expression, and stress response through multiple enzymes. PARP is extremely important, and as you will see later, as sirtuins. So this is complicated, but it is so important because what PARP does, PARP repairs damaged DNA. That's it. So we need PARP. How can we get PARP? We can get PARP from NAD. And you see, look at this. PARP is often considered our guardian in genome integrity. So it's like, again, it's one leads to the other. Now, I showed you previously the mitochondrial function through oxidative TSA cycle, oxidative phosphorylation, where there is a participation of NADP in production of ATP, as well as the synthesis of mitochondrial glutathione. NAD is reduced to NADH, and then through NADHP. And again, this is so important, as I said, to support mitochondrial oxidative phosphorylation, to generate ATP, to generate our energy. Know this, know this, know this, know this, no energy. This is a complicated slide. This is maybe too much. Sorry, I thought that I actually hit it before. NAD and sirtuins. If NAD, so if DNA damage is not repaired, the PARP activation is persistent, declining cellular NAD, and this can impair sirtuins, altering our epigenetic chromatin structure, and eventually mitochondrial and cellular metabolism. Sirtuins. Sirtuins are essential part of our life. How many sirtuins there are? Now look how new they are. 2001. There are 7 sirtuins. Sirtuin number 1, extremely important in our DNA. It modulates that famous tumor suppressor gene P53. Since sirtuin activity is known to decline with age, likely as a result of declining NAD, the loss of sirtuin-mediated tumor suppressor function may be a critical step leading to tumorigenesis. Sirtuin 2. Sirtuin 2 has been demonstrated to deacetylate. To deacetylate meaning, deacetylate, it basically quiets down DNA, quiet down those histones, meaning that those genes have to rest. They don't have to be active all the time. However, it also upregulates something that enhancing NADPH production. So, and this cannot act without NAD. None of the sirtuins function without NAD. Sirtuin 3. Now, mitochondrial sirtuins has been identified as an essential mediator of different processes and adaptive responses to energetic challenges in the brain. So it deacetylates or turns down, lets this kind of a gene to rest. And then it works with superoxide dismutase, and we know the function of superoxide dismutase. The Sirtuin 3 modulation has been implicated as an oncogene and tumor suppressor in cancers. Sirtuin 4. Sirtuin 4 has been identified as a deacetylate that acts as a tumor suppressor in multiple malignancies. Sirtuin 5 is unique. It acts predominantly as lysine desuccinylase. Whatever it does, it modulates the numerous mitochondrial metabolic enzymes. Sirtuin 6, it modulates DNA. Here we go, our famous telomeres. And Lapshes were mentioning sirtuins and telomeres at one of the training calls. So Sirtuin 6 modulates DNA repair in telomeres and participates in repair. Now, by recruiting this PARP1 to repair the DNA breaks, how important it is. Very important. And the last one. So that one has been implicated in aging and cancer physiology, but is relatively poorly characterized. NAD and importance of it. The multifaceted roles of NAD on energy production, DNA repair, antioxidant defenses, and many other signal transducing pathways make this metabolize a gem-like substance for cell survival and homeostasis. Maintaining NAD at optimal levels throughout lifespan might be essential to prevent metabolic maladaptation, age-related neurodegeneration, and cancer formation. That is why I think it is so important for us to teach and spread the word of NRF2 activation, mitochondrial health via NRF1 activation and NAD upregulation. Now, let's talk about some of the dosages, niacin doses. So if you know, probably if you look at the NAD bottle and you see the doses of our cuprous niacin, you see how much lower the dose is. Our is in micrograms. So this is adverse effect of nicotinamide are rare, and they occur with oral doses over 6 grams a day. That may cause nausea, vomiting, liver toxicity, headache, fatigue, and dizziness. Now, we do have a niacin in Axio, and some people experiences a flushing effect or the niacin flush. Nicotinamide does not cause flushing. It seems like NAD also shows to be a free radical scavenger. It can be involved in the calcium signaling pathways leading to apoptosis, which is great. Safety first. Nicotinamide is not carcinogenic by itself at doses more than 300-fold above requirement, administered in mice throughout their lifespan. So, and these are some doses that I have learned through my literature search. Copper, what I have found, and I don't know whether it's right or wrong, but again, through the literature search, that copper is a transporter. So we need it to transport that niacin, amongst other functions, actually. This was done, this is a human study. So administration of 100 milligrams per day of niacin to 2 volunteers protected against lymphocyte DNA strand breaks induced by some free radical elements like xanthine oxidase. The supplementation increased NAD concentrations by nearly 5 times baseline and significantly reduced oxygen radical-induced DNA strands breaks in those lymphocytes. So we don't know a lot of things. So we know that NAD precursor treatment is emerging as an efficacious intervention for the prevention of numerous age-related pathologies, and it has a tremendous therapeutic potential. We as healthcare professionals, we need to be safe, and we just need to study a lot. So this is what, guys, I wanted to share with you. I don't know whether it was helpful or not, but I hope it was, and probably I can open the discussion or maybe answer a few questions. So I will stop recording, and